Hello guys, welcome to your next installment of Allied Health 102, Intro to Phlebotomy. Okay, and as you already know, I'm your instructor, Melissa Shepard, and we're here today to talk about the cardiovascular and lymphatic system, and that's chapter 7. And remember, our first test is going to come from chapters 1, 6, and 7. And also remember, you should already have watched your first PowerPoint presentation. You should have, on chapter 1, you should have read chapter 1 in your textbook and filled out your review guides for chapter 1 that are located in your black binder. Then you should have moved on and read chapter 6 in your textbook, watched the PowerPoint presentation on that chapter, filled out your chapter review guides, and you should be studying those chapter review guides for chapter 1 and 6, and you should be reading chapter 7. You'll watch these PowerPoint presentations, fill out those review guides, and it'll lead into our first test. Okay, also remember, we just want to keep you up to date. You know, we use that tentative course calendar as a guide to tell us when our quizzes are coming up and when they're due, when our tests are coming up and where they're due, and kind of where we are in things in both lab and lecture. And remember, due on October the 13th will be our research paper. And although we didn't download it from Canvas, right, because of COVID, I handed all that out to you on your first day in lab, and you placed it in your black binder. Now, there's topics in there, topics relating to phlebotomy. And you'll pick one of those topics, and then you'll find 3 references, 3 solid, good references. And one of those needs to be from the BIPSI library. Now, I know you're probably thinking, you know, COVID's going on right now. I can't get on campus. How can I go to the library? I'm going to have a set of videos available to you on your Canvas course that are a lot of great tutorial videos showing you all the fantastic things that are going on in our BIPSI Learning Commons library and ways for you to access them and be able to do curbside pickup for books, for you being able to get information, tutoring, videos, all kinds of things. And so I'm going to link those videos inside this Canvas course, and you'll be able to find them. We'll talk about them in lab as well and in our live conferences for lecture. And you'll be able to have those resources that you need to be able to write your research paper. And we'll talk more on our research paper in lab class. But remember, it's due October the 13th, so you need to start narrowing down your topic, and we'll start looking for references. Okay, these are our learning objectives, right? I'm not going to sit and read each and every one of the learning objectives. The thing you need to know about this, first and foremost, is that these are the things that when we finish discussing chapter 7, and you finish reading chapter 7 in your textbook, which these learning objectives are located in the textbook as well, and you fill out your review guides and you start studying those review guides, you will know all this stuff. Everything from being able to trace the flow of blood through the cardiovascular system to locating and naming the veins and arteries most commonly used for phlebotomy procedures, and all our other objectives, including talking about hemostasis. So we're going to start with the cardiovascular system, all right, as a whole. So the 4 functions of our cardiovascular system are transportation, regulation, hemostasis, and it works as a distribution for the nutrients, right, that we get from our digestive tract, from breaking down of the food and drinks, beverages that we drink, water, and those things go through our digestive tract and they're absorbed into our bloodstream, right? And then they're taken all over the body where they're needed, right? So regulation, our cardiovascular systems helps us regulate our blood pH, our electrolyte balance inside our blood, and our body temperature. And then we've already talked about homeostasis, right? Homo, like a Homo sapien, our body. A homeostasis is how our entire body stays in that steady, normal state, right? Well, hemostasis is talking about that balance of blood, specifically dealing with the coagulation when we talk about how our body's able to balance that liquid state of blood as it flows through our vascular system, but being able to clot when need to, okay? And then our primary components of our cardiovascular system, and they're made up by the pictures you see down below there over to your right. That beating heart, right? That muscle, okay, that works diligently for us, pumping the blood through our blood vessels, which is our next little picture, and you see the cutaway, and you see the cells inside those blood vessels. And then last, that circulating blood that circulates through those blood vessels, and you'll see that picture of someone there with their arteries and veins and how they flow all over your body to the tips of your fingers, and they take oxygen and other nutrients to every cell and tissue and organ in your body. They pick up the waste and then they do it all over again. So we're going to start with the heart, right? And as I said, it's muscular organ, right? And we've heard this before. If you close up a man's fist, that's about the size of the heart, right? Okay, one of the things you always need to remember, we always want to stay healthy, right? We always want to stay healthy. And one of the ways we can stay healthy, and we'll go through some of the other things that you can do to stay healthy, is that we want this to be a strong muscle, right? And so we need to work out our heart. That's why it's important to get out there and do brisk walks every day and get your heart rate up a little bit so you can make sure that that muscle is strong. Because what happens, you've seen people that have a cast on their arm, right? They take the cast off, and because they've been unable to use that arm, the muscles in that arm are quite a bit smaller than the muscles in the other arm, and they're weaker, right? Well, we don't want a weak heart muscle. It's vitally important, so we want to work that heart out, whether you're jogging, running, walking, riding a bike, to make sure that you keep that muscle fit and strong, right? So now let's move on and start talking about the parts of our heart, all right? So we have two parts of the heart, all right? We have the upper section of our heart, and you'll see in that picture with the atria, and they're separated by the interatrial septum or wall, okay? And then we have those bottom parts of our heart, the ventriculars, which are separated by the intraventricular septum or wall, right? And as we go into this, you'll see and understand why it's important to have these separate parts, right? So when we're still talking about the heart, we're talking about how the blood flows in and through the heart, right? And this is a fantastic picture with arrows that show you outline of how the blood comes in and flows through the heart. And this is something you're going to need to know, guys. So start using this picture and this information to study now. So the largest veins that enter the heart, they enter at that right atrium. And you can see those areas from that inferior vena cava and that superior vena cava, where that blood is coming into that heart and it's dumping into that right atrium, right? So your heart beats and it pumps blood in through those veins and into that right atrium. So let's talk about the flow through the heart, okay? Because this is something you're going to need to know, okay? So that, like we talked about, it comes in through that inferior and superior vena cava into that right atrium, right? Then the heart beats again, right? It pumps again. And then it goes through that tricuspid valve and it enters the right ventricle, okay? Now we need those valves there so that that blood doesn't backflow back into the right atrium, right? Because we brought this blood into the heart that is low in oxygen, high in waste like CO2, and we don't want it going backwards, right? We need it to flow through this process, right? So that tricuspid valve, when that heart pumps, it opens up that tricuspid valve, takes the blood that was dumped in the right atrium, sends it into the right ventricle. Then it passes through the pulmonary valve, and then it goes to the lungs through that pulmonary artery. All right. Now remember, a lot of people try to remember your veins and arteries because you say the veins are not oxygenated and the arteries are oxygenated. But a better way, because you can get messed up on two different places, and this is one of them. Okay. Your maternal blood flow is another issue, and then you can get messed up there. But we remember that if blood is going to the heart, it's a vein, right? If it's going away from the heart, it's an artery. So we see that it's in the right ventricle now, right? The heart beats, and it passes through the pulmonary valve and goes to the lungs through the pulmonary artery, right? Because even though it hasn't made it to the lungs yet to exchange gas and be oxygenated, it is leaving the heart. So it is an artery. Okay, so it goes to the lungs, it does its gas exchange, dumps off its CO2, picks up its oxygen, and now it's leaving the lungs through the pulmonary vein, right? Because we're going back to the heart, right? We came into the heart, we didn't have any oxygen, we left the heart to go to the lungs, that's why we were in a pulmonary artery. Now we've been oxygenated, we're coming back into the heart again through the pulmonary vein, right? So away from the heart is an artery, going to the heart is a vein, right? So we're going to leave the lungs, we're oxygenated now, come in through that pulmonary vein, and we're going to enter on the other side of the heart at the top in that left atrium. The heart beats again, it passes through the bicuspid valve, and then enters the left ventricle, okay? Then it passes through the aortic valve, and it leaves the heart through the aorta, okay? There's that big artery up there at the top. You see now it leaves, and now it's going to take this oxygenated blood, and it's going to deliver it out to all the cells and tissues and organs of our body. So practice this, go through it, because you will need to know how the blood flows through the heart. You're going to need to know about the right atrium and the left ventricle. You're going to need to know about the tricuspid valve. And you're going to need to understand about the pulmonary artery and the pulmonary vein. So here again, and you'll see these on all my PowerPoint presentations, these are what I refer to as critical thinking or higher learning activities. Now what these are for is if we were in the classroom, you would get a chance to raise your hand or shout out loud, or maybe we would get a piece of paper out and everybody would write down their answer, because these can be used for you to see if what you're going through, are you putting it together, or are you learning it? You can use these critical thinking slides as ways to study at home yourself, okay? And again, it's like I told you guys, you will be successful in this class, you will have a really good grade, and you will learn this material and be able to carry it forth through all your other classes if you will take the time to read your chapter, watch the PowerPoint presentations, fill out your review guides and study them, and then use things like these slides to practice and see if you're getting this information. Okay, so we're talking about the heart, right? So we got a systemic circuit and we got a pulmonary circuit, right? Okay, so remember on the pulmonary is lungs, right? Okay, so that will help you understand that the pulmonary circuit is the blood that goes through the lungs. And systemic, think of systems, right? Organ systems out in the body, right? So the systemic circuit is the blood that goes out and through the body. We have a systole contraction and a distal relaxation, okay? So you think of the contraction and relaxation of the ventricles, right? And we're going to go into that more when we talk about taking your blood pressure, okay? So here again is another one of our slides. So use these as a tool to practice and see if you can answer, because you can always come back to these slides and flip through them and go to these to test what you're learning after you study your review guides. Okay, so if we were in class, we would do this right now, but we're obviously not there right now, but you will do this in a lab, okay? But you'll hear me say a lot when we're in lab, not when we're drawing blood, when we're doing other things like taking blood pressure, how one a valuable tool you have is to be able to visualize things, to be able to visualize and run that process through your head to make sure you're doing everything from A to Z in the correct order. You need to, when you're feeling for your veins, you need to be able to visualize how big the vein is, which way it's running, how deep it is, so that as you go to stick your needle into your patient's arm at the antecubital fossa of their arm, you will be able to visualize exactly where you need to go. Visualization is a great tool you can use, okay? And right now, as you're sitting here listening to this, visualize the things you need to do in the order you need to do them in. And everybody sees how important washing hands is now, but it's always been important to allied health and nursing, okay? Always. Because we know that there's other things other than COVID-19 that are out there that can be spread from patient to patient that can make people sick, make people disabled, and kill people. And one of our first lines of defense is hand sanitation, okay? Hand hygiene. So washing your hands is vitally important before you do anything dealing with the patient. Okay? So you let your patient sit down for a few minutes. You want them to sit down and relax, right? During that time, and you'll hear me talk about turnaround time and being able to kill two birds with one stone and how to maximize your time and being organized. That is vitally important in any form of healthcare. So you wash your hands, get your patient seated. While your patient is sitting there for their 5 minutes, you assemble all your equipment, right? You need an appropriate cuff size. It needs to encircle at least 80% of the arm. You always, always, always want to identify your patient, because you would not want to take the blood pressure of someone, write it down in a chart, and you didn't properly ID your patient and you wrote the wrong blood pressure down in the wrong person's chart, okay? So vitally important to always identify our patient with 2 different measures to make sure that whatever you're doing to your patient, you're doing it to the correct person, okay? Then you want to support their arm on a flat surface, apply the cuff around the arm with that arrow, and it'll be on the cuff over the brachial artery, okay? You want to clean your earpieces, always clean your equipment, okay? Place it in your ears, place the amplifier, the round disc area, over that brachial artery, inflate to approximately 170 millimeters of mercury, right? Open the valve slowly, letting the air out of the cuff. The systolic blood pressure is the point at which the first pulse sound is heard. And the diastolic blood pressure is the point at which the last pulse sound is heard. Then you would record your readings, repeat it one more time to make sure you're getting the correct, until you get really good at this. It's always good to repeat it a second time just to make sure your numbers are coming real close to one another. Then you're going to always wash your hands and your equipment, and then make sure you put everything away in the proper place. And we will be doing this activity in class. Still talking about that heart, you know, that heart beats, as you saw, all day, every day, 24 hours. So that fluid surrounding that heart cuts down on friction. It keeps it moving and lubricated, okay? That pericardium, right? That pericardium, that is that double layer of fluid-sealed sac that encloses your heart, right? Now, these are just words that we need to know the definition, and we need to understand. We need to know what the pericardium is, okay? What is a myocardial infarction? It's a heart attack, right? You hear people saying MI. This is where, for whatever reason, those blood vessels, because the heart's a muscle too, right? It has blood vessels leading to it, right? So that oxygen and nutrients can be delivered to the cells of the tissues of your heart muscle. And if those become clogged up for whatever reason or fail, they may not be able to deliver the oxygen and nutrients to that part of the heart, and that part of the heart muscle could die. And that's when we talk about an MI or myocardial infarction, heart attack. So the instrument, and I'm not going to try to pronounce this, I can, but it usually takes me about the 3rd time to do it, to get it exactly right. Some people can get it right off the bat. If you need help with it, there's always stuff online where you can see how to pronounce it properly, and I don't want to butcher it twice before I get it right the 3rd time. But it's a blood pressure cuff, right? And this is an example of a normal blood pressure. It's 120 or less and 80 or less for your bottom number, right? So 120 over 80. So now we're going to talk about vessels and circulation, right? There's 3 kinds of blood vessels that exist in the human body. We have arteries, veins, and capillaries, okay? Now, if you look at these, there's different diameters of these different vessels, okay? And so the inside of the blood vessel is called the lumen, okay? That's the inside, when you look on the inside, okay? And as we age, and other factors play a part in this, like your diet, whether you exercise, if you smoke, if you're overweight, all these things can speed up the process of what we refer to as hardening of the arteries or atherosclerosis, okay? Age is a factor, but other things play a factor in it as well. If you don't smoke and you exercise regularly and eat a healthy diet, then it may take a little bit longer for you to start experiencing any hardening of the arteries or atherosclerosis. If you, at a very young age, you've smoked for years and you eat a very unhealthy diet and you're what they refer to as a couch potato or a sedentary, and you don't exercise or move around hardly at all, you may start to see signs of hardening of the arteries at a much younger age, which you don't want. So you think about this. So we all heard about plaque, right, in our arteries and stuff. And people will talk about if your cholesterol is high and if you eat a high fatty diet all the time, you know, you can build up plaque in the arteries. Well, the issues with this is what ends up happening is when you're young and you're healthy, the inside of your artery walls are nice and clean and smooth, and the blood just flows through it really, really good, right? But if you have inflammation due to maybe diet or stress or smoking or any of these things that causes inflammation inside those blood vessels, the insides of those walls, they're not doing good. They're inflamed, and then they can start to get sticky and build up plaque, right? And then as the cells move through them, some of the cells can get stuck on this plaque, right? And then it narrows and narrows and narrows, and it could result in a stroke, right? And then you can see blood supply to the brain could be reduced. You could have, like I talked about earlier, a lack of blood supply to parts of the heart. And then when you're not getting blood supply either to the brain or to the heart, it starts to die, right? And that's when you're talking about a heart attack, right? So arteries, right? And like I said earlier, be careful with the arteries are oxygenated and veins are not. But for the most part, okay, arteries are highly oxygenated vessels that carry blood, this is the important part, away from the heart, right? And then they branch out into smaller and smaller vessels till they get down to the smallest one, which are almost single-celled lined capillaries, okay? Arteries are usually brighter red in color, and they have a thicker elastic wall because they have the pressure of that heart behind it. That's why you can feel a pulse in arteries versus veins, because it's going away from the heart and it's right behind the beat of that heart to send it out to all the cells of your body, even at the tips of your fingers and your tips of toes. So that's why you get a pulse in there, and due to that, the walls of those arteries need to be a little bit thicker to take that pressure, okay? The three most common used arteries for arterial puncture are your radial artery, which is found in your wrist area, the brachial artery, which is found in your arm, and your femoral artery, which is found in the top part of like the groin or bend area of your leg, okay? So of these three, the most commonly used artery for arterial puncture is the radial artery. And we'll go more into this about the Allen test, and we'll talk about this in the lab, and we'll talk about, you know, how some areas of your body have arterial blood flow. They have two different arteries that supply blood flow to that particular area, so you can use one for, it's better to use one for arterial puncture, because if something happens to that one, it doesn't stop blood flow to that area. So we'll get into all that and talk about the radial artery and the Allen test later on. So now moving on from arteries to veins, right? Blood is carried towards the heart by the veins, right? Veins have one-way valves that allow the blood to flow against gravity and prevent the backflow. So that's why you don't really have that pulse, right? Because it doesn't have as much of that, it doesn't have that pressure behind it, right? Because the pressure pushed it out there, and now gravity is just going to help it flow back to the heart, right? Okay, but we don't want it to backflow like when we talked about the valves in the heart. Well, there's those small valves in our veins that prevent the backflow, okay? And we arteries don't need those, right? Because it has that pressure behind it, okay? So they're not quite as thick as arteries because they don't have to hold up to that pressure behind it as much as arteries do. Okay, so if you look at this arm picture here, down at the bottom on the right-hand side, if you hold your hand out right now, your arm out right now with your palm facing up, right where you're on the back side of where your elbow is, that is the antecubital fossa of the arm. And the antecubital area of the forearm is the most commonly used area for venipuncture, right? And the median cubital vein is the best best for venipuncture because not only is it the largest vein, okay, but it's the best anchored. Not the median vein, the median cubital vein, okay? And then you'll see from this diagram that we have a cephalic vein, a basilic vein, right? And you will need to know these veins and be able to identify them where they are on a photograph, such as the one you see here. So go ahead and make a habit out of starting to study these veins. And could you, if you white it out, that's the cephalic, that's the median cubital, could you go and look at this picture and be able to write in which vein was which yourself, okay? So again, these are our critical thinking slides. You can use them as a way to study and practice to see if you're starting to piece this stuff together and be able to answer these questions that will most definitely be on a test you see. Okay, so arteries, veins, capillaries, okay? Capillaries, excuse me, like I said before, are the thinnest, okay? And they connect your arteries to your veins. So, and it makes sense, right? That blood leaves the heart through that aorta, right? It's got that pulse behind it. It pushes it out all the way to the tips of your fingers or the tips of your toes out to the capillaries, right? And then the capillaries hook back into the veins, right? So we can go back to the heart, right? They're like one layer thick. So the capillaries being that thin, they're able to permit the exchange of gases, picking up your CO2 as a waste product, taking it back, and then inside your lungs, down in there, you'll have those little capillaries that let the gas exchange where it drops off the CO2 and picks up the oxygen, okay? A small artery is called arteriole, and a small vein is a venule, okay? And you've got to think of capillaries as they're meeting and they're combining together, they're hooking together your arteries to your veins, then it's kind of a mixture of arterial and venous blood. But because, and if you think about this, because there's more pressure behind that blood in the arteries, they're going to get a little bit more of their blood into that mixture than the veins could get. So the capillary blood does resemble the arterial blood in makeup a little bit more than venous blood, but it is a mixture of two, and I always compare it to brackish water. So if you think when freshwater lakes or rivers or streams run into the ocean, right? It's going to be that area in between where it's not really fresh water any longer, but it's not quite ocean water as well, but it's a brackish type of water. That's what you see with this capillary blood. Okay, now talking about the blood itself. Because remember, when we were talking about those three components of our cardiovascular system, right? We were talking about the four jobs that they do and then the three parts of it. We talked about the heart already. We talked about the blood vessels. Now we're going to talk about the blood that flows through those vessels, right? So if a blood specimen is taken, right? We use a vacutainer setup, we go into our vein and we draw some blood and it goes into our vacutainer tube. So here's a picture of a vacutainer tube, right? We drew some blood in it, okay? Now, about 55% will be the plasma, that part that you see at the top that's that yellowish, amberish color, right? That's going to be the plasma. Now, of course, this is after we have spun this tube down in centrifuge, right? And then down at the bottom, past that yellowish will be another 45% will be formed elements or cells. Now, at the top, it could be anywhere between 55 to 60% plasma, okay? Then you're going to have approximately 1% in what we call the buffy coat, that's white cells and platelets. And then down at the bottom is anywhere between 40-45%. Now, this is in a normal, healthy individual, would be those packed red cells once we spin that down. Now, the plasma portion contains approximately 90% water and 10% solutes. Like our body itself is mainly made up of water, that's going to be the same as the plasma portion of our blood, okay? The circulating blood cells we have different kinds, right? We have red blood cells, RBCs, erythrocytes. These are different words for the same thing, right? We have white blood cells, which can be referred to as WBCs or leukocytes, and then we have platelets, which are thrombocytes, okay? And you'll see the little picture of the erythrocyte, the leukocytes, and the platelets to the left of our tube here. Okay, so this is a little diagram, and that big cell circled at the top, that's our stem cell, okay? Now you think of a stem cell as a cell that doesn't know what it wants to be when it grows up, right? Okay? It's where all the cells come from. It's the mother of all other cells, right? And hemopoietic, okay, blood-forming tissue, such as the bone marrow, is the one that gives birth to these stem cells. Okay, so you think and it's not just in the bone marrow, but mainly in the bone marrow, you have this special hemopoietic tissue that forms the stem cell, right? And you see this line, okay? So over here, you have the one circled at the top, that's your stem cell, right? Again, this is a cell and it doesn't know what it wants to be when it grows up. All of them start out as a stem cell. So if you see the column that's over there to your far left, okay, that first one right there to your far left, that's a ruboblast. The first, now it knows it wants to be a red cell, okay? It's not a mature red cell yet, okay? It's not ready to be dumped into your bloodstream yet and do its job, but it now knows that it wants to be a ruboblast. If you see the far right line, okay, that first cell that comes off the stem cell there to the far right, that's a megakaryoblast. Okay, it now knows it wants to be a platelet. Now, it's not a mature platelet yet. It's not dumped into the bloodstream yet, but it now at least knows it wants to be a platelet. Okay, so the blast, or the immaturest line of each of the individual cells, whether it's a ruboblast, okay, it's that, if you hear blast at the end, then it knows that it wants to be a specific cell, but it's in its most immature form, okay? And then it goes all the way down until you get to the erythrocyte, okay? Okay, again, these are another one of our study activities. You can write these all down, and then you can see if you're getting your correct answer. And you can go back through these PowerPoint presentations. You can fill out your review guide. This information is loaded up everywhere, and we will discuss this in our live chats and in our laboratory setting. So let's talk about our cells now, okay? So we talked about stem cells. They all came from stem cells, right? So now we're talking about erythrocytes, right? So just a little bit of information on these RBCs. They're very, very small, 7 microns in diameter, okay? And prior to reaching their maturity in the bone marrow, they lose their nuclei and become enucleated and are a biconcave disc. It's like somebody took it and pressed in the center, right? It's thinner in the center, okay? It almost looks like a donut, a red donut, when you see it on the microscope. There's about 5 million RBCs per cubic millimeter of blood. Very small, a lot of them. Within each mature RBC are millions of hemoglobin molecules, okay? So each one of these molecules is capable of carrying 4 oxygen and 4 carbon dioxide molecules, okay, for each one of those hemoglobin molecules. That's a lot of oxygen carrying power, right? So erythropoiesis, okay? Erythropoiesis is the term that refers to the production of RBCs, okay? So how does that happen, right? How do we produce our red cells, okay? We know that we have stem cells. Those are cells that don't know what they want to be inside our bone marrow, right? Okay? Well, think about this. It's usually a hormone that triggers things, right? We talked about insulin, you know, telling our body to take the excess glucose out of our bloodstream and store it, right? Well, erythropoietin, that's the hormones, and it's produced in your kidneys, that triggers the erythropoiesis process, okay? So when your body is not receiving enough oxygen, okay, then the kidneys are stimulated to produce erythropoietin, right, the hormone, right, which then in turn activates your bone marrow to produce more RBCs. Now, one way you can do this naturally and normally, right, and the more red cells we have, right, the more oxygen we can get to our tissues, right, the healthier we're going to be. We'll be more oxygenated, right? We want to do this. One way we can do this is by exercising. Because you can imagine, right, when you first take on an egg, you go to your doctor and you want to be cleared to get out and exercise. And first you say, I'm going to run. So you run for a short period of time and then you're breathing heavy and you have to walk. And then you can run a little further and a little further. And to the next thing you know, you're running a mile or you're running a 5K, right? Well, as you're doing this, you're forcing your body also to produce more red cells, okay? And it makes you healthier that way, okay? But then there is an unnatural way to do it. So a lot of you have heard about sports cheating, right? Taking steroids or banned substances. Well, that's how a lot of athletes such as world-class cyclists, marathoners or sprinters or runners, people who need a lot of oxygen delivered to their cells. If you're riding a bicycle at high rates of speed up a side of a mountain, you need as much oxygen to those muscles and cells as you can get. Well, one way these athletes do this in a non-natural cheating manner is to take something that mimics erythropoietin that tells your bone marrow to produce more red cells, because of course the more red cells you have, the more oxygen you can carry, which means it may give you a slight advantage as a cyclist or a runner, and that's cheating. But that is something you guys may have heard of in the past. So moving on from that and talking about erythrocytes, right? So the process of maturing in the bone marrow from that stem cell, right, that we said is the cell that doesn't know what it wants to be when it grows up yet, right, all the way to a mature RBC, right, takes a few days for this process to occur. And each stage has a name, okay? And it processes from that ruboblast. Remember when I talked to you and this shows this, each line of cells, right? So first you're that ruboblast, right? You go from the stem cell to the ruboblast. Now I know I want to be an RBC, so now I know what path I need to take. So then I have to go through several other stages to get down to an erythrocyte, okay? And the name of the stage that's immediately prior to that mature RBC, and you'll see it because you see it losing its nucleus. If you see that one right before you get that biconcave erythrocyte down at the bottom on this slide, on this image here, you see that little purple thing coming out of the cell, that's it losing its nucleus. That's that reticulocyte, okay? So the lifespan of RBC is about 120 days, okay? So it's, it circulates through your body doing its job for about 120 days. Then it starts to fragment off, right? It breaks up and is released into your bloodstream, okay? And then we talked about how our liver cleans our blood, right? So those fragments of that dead and now red cell are removed by your liver, your spleen, and your bone marrow, okay? So as your body breaks down that hemoglobin, it's broken down into two different things, iron and bile pigments, okay? And pigments are colors, right? The pigment of your skin, colors, right? So it's broken down into iron and it's breaking down into bile pigments. So the bone marrow itself will reuse that iron to make new RBCs, okay? We recycle it, okay? And then those bile pigments, when we excrete our feces, it is one of the things that gives that color. So it's excreted into the intestines and then out through our feces, right? So that's kind of how that whole lifespan of that RBC works. So now we're going to move on to blood typing, antibodies and antigens. Okay, guys, people get so confused on this. We have missed questions on tests. So I really want you to focus in on this, and you may need to watch this portion of the video more than one time so that you can get it, okay? Because we're going to talk about two different systems, okay? And the first one is our ABO blood group system, okay? And if you are a human, you have one of the four blood types, A, B, AB, or O, right? O is the most common blood type. Then in order, it's after that it's A, B, and then AB is the least common, okay? The differences in blood types are due to the presence or absence, okay, of these antigens that are on your red blood cells, okay? And these antigens are made up of protein molecules, okay? And they're actually right there on the surface of your RBCs. They're attached to the surface of the RBCs. And then with that, you have these naturally occurring antibodies to the antigens that are located on the blood plasma. They're located in the blood plasma, I'm sorry. So the antigens are on the surface of the red cells themselves, attached to them, okay? And then the antibodies are in the blood plasma circulating around, okay? So that's the first thing you need to make sure you understand, is that the antigens are located on the RBCs, the surface of the cells, and the antibodies are floating around in that blood plasma, okay? So I'm going to go through this, but you may need to listen to this several times so you can make sure that you're getting it together, okay? So let's start with people with blood type A. If you are blood type A, then you have those A antigens on the surface of your RBCs, okay? And because you are A, then you're going to have the opposite. You're not going to have A in your plasma, right? Because let's remember, the second thing you need to remember is A, if you have A and it comes in contact with anti-A, it's against A, right? So if you're against something, if you're on a football team and you are against the other team, you're going to try to go tackle them, right? Okay? You got to think of that, anti. Okay, so if you have the A antigen on the surface of your red blood cells, then you are type A. Okay, but you will not have anti-A in your plasma. Okay, you will have anti-B antibodies in your plasma. Okay, and if you are type B blood type, then you will have, it's just the opposite, right? So follow through this, guys. It's not hard once you really piece it together. If you're a blood type B, then you have those B antigens on the surface of your red cells. And of course, you would not have anti-B in there, right? Because then it would attack it and destroy your red cells. So you're going to have anti-A. A has anti-B. B has anti-A, right? Now, okay? So individuals with type AB, they're going to have both, okay? So you're going to have both A and B on the surface of your red cells. So think this through, guys. If you have both A and B on the surface of your RBCs, then you want neither anti-A or anti-B in your plasma, right? Because if you had either anti-A or anti-B in your plasma, it would attack the A or the B on your red blood cells and destroy your red blood cells. So if you have A and B on your red cells, then you have neither A or B in your plasma, okay? Now, O. O means you have neither A nor B on your red cell. So if you have neither A or B on your red cells, then what you now have is both A and B antibodies in your plasma. Okay? Makes sense, right? Whatever type you are is what you have on your red cell, and you have the opposite of that in your plasma. Okay? So if you're type AB, you have both A and B on your cell, so you wouldn't have either in your plasma. In O, you have neither on there, so you have both in your plasma, right? So since AB people have no antibodies in their plasma, right, then they're the universal recipient. Because no matter what blood they get, they have nothing in there to attack it, right? And O is a universal donor for the exact same reason, opposite, right? Because I can give you O blood, right? So because O, the universal donor, since they don't have anything on their cells, then it wouldn't matter what was in the plasma because there's nothing to attack, okay? So remember, the reason AB is a universal recipient, they can receive any blood, is because they have neither anti-A nor anti-B antibodies in their plasma. So no matter what's on the cells themselves that they're getting, so they can get any cell, there's nothing in their plasma to attack, right? And it's the reverse of that with the O, right? Because O has nothing on their cells. So if you get the O cells, it doesn't matter what you have in the plasma because there's nothing on those cell surface to get attacked. Okay? Makes sense, right? Okay. Here's a way for you to see if you're getting this stuff right. And if you can't answer these correctly, you need to go back and watch that other slide again until at one point, I promise you, it will click. If you take the time to go over it several times and then test yourself to make sure. If not, I'm telling you, it's not going to click quickly for most people. And you're going to have that test and you're not going to get these questions right. So use this as a tool to see if it's clicking with you yet. And if not, then put in the time to go back and study it until it clicks. Because that's what it's all about, guys. If you're not willing, nothing good ever came easy, okay? And if you're not willing to put in some time and effort and work, you're not going to learn this stuff. It's not going to be easy. If you do what I'm telling you, when you sit down to take that test, then you're going to know the information and you're going to pass the test. If you don't put in the time and the hard work you need to put into, when you get that test, you're not going to do well. And the only person you're going to be able to blame is yourself for not setting aside the time you needed to put in the hard work you needed to make sure that you were successful. So use these slides to see if it's starting to click with you. And again, you can pause these to make sure that you're getting the right answers so that you will know that it's clicked with you now and you do understand. So this is a clinical alert, okay? Because as I've told you guys, there's lots of, in healthcare, there's identifying your patient is vitally important. Whether you're identifying your patient to send them for some type of diagnostic testing, including laboratory tests, which may include cross-matching of blood because they're going to get blood for a surgery they're having, or if they're in an accident or have an injury. Then there's also identifying your patient if you're taking their blood pressure or something like that. Identifying your patient is important. Identifying that the person that you're drawing this blood on is the correct person and making sure you're labeling your tubes properly, because if not, you could have blood transfusions could cause a reaction that could cause you could damage their kidneys for life, you could cause disability, and frankly, you could cause death. Okay, because what happens is, so I'm a type A, right? And I got A antigens on my cells, and I got anti-B floating around in my plasma. But you give me somebody that's type B blood. I'm not type B, I'm type A, right? Type B blood has what floating around in its plasma? Anti-A. So now I've gotten the blood from a person who's blood type B when I'm a blood type A. They've got anti-A floating around in their plasma. Some of that gets into my bloodstream, attacks my RBCs, and it starts to hemolyze them or rupture them or clumping them all together, agglutinating them, which then could clog up small blood vessels. It could damage my kidneys, my lungs, get a clot in my lung, and my heart, and my brain. It could lead, like I said, to just some type of disability and even death. That's why it is vitally important that you properly ID your patient and that you properly label your tubes. That's why we never label any tube before the blood's been drawn. Okay? So doctors, that's why doctors do cross-match testing, but still, if you cross-match testing with the wrong tube of blood, it could still turn into a problem. So properly IDing your patient is vitally important. So still talking about erythrocytes, okay? So blood types are determined by the ability of an antibody to cross-react with the antigens on the surface of the red blood cells, right? We've been talking about this. So how do we test for that? So we take a drop of our blood, right? So if our blood cells, we put a drop of blood and then we put a drop of anti-A and we mix them together. If they agglutinate, which is clump up, make little clumps, then our blood type is A, okay? If we put a little drop of our blood and a little drop of anti-B and mix them together and they clump up there, then our blood type is B, okay? Because you're going to always do 3, right? It's going to be the anti-A, the anti-B, and as we talk about later, anti-D, okay? So if you have them all out there and it clumps up in the A, but it doesn't clump up in the B, then you're blood type A, okay? If it clumps up in the B, but it doesn't clump up in the A, then you're blood type B. If the cells agglutinate with both the anti-A and the anti-B, then we're type AB, and if it doesn't agglutinate in either one of them, you're type O. You guys are going to need to know this for the test, okay? So make sure you keep going over this until it clicks. And here are some slides that if you pause these and see if you can answer each one of them correctly, you will then know if this stuff is starting to click with you, and then when you sit down for your test, you'll be able to pass it. So now we talked about, remember at the beginning I told you there was 2 blood group systems, and the ABO blood group system was the first one, and it would determine whether you were type AB, or O, right? Now we're going to talk about the RH factor, okay? So if you're RH positive, that means you have the RH factor or D antigen, right, on the surface of your RBCs. If you're RH negative, then you do not have that D antigen on your red cells, right? Now, so you think about this, okay? So if you have the A antigen on your red cell, right, then you're going to have anti-B in your plasma, right? Because your blood type is A. So you have A antigen on the surface of your red cells, and you have anti-B floating around in your plasma, right? If you're RH positive, in other words, if you're A pos, then you're going to have the D antigen on the surface of your red cells as well, okay? But if you're A negative, then you will have that A antigen on your red cells, but you will not have the D antigen on your red cells, okay? So how can this happen otherwise, right? Okay, RH antibodies are present, okay? So we know you do or you do not have the antigen, right? So we know we're talking about antigens which are on the surface of the cells and antibodies which are in the plasma. So RH antibodies are only present if the body has been exposed to RH positive red blood cells, either accidentally by transfusion, okay, or during childbirth when an RH negative mother is carrying an RH positive fetus, okay? So in other words, if you are A negative or B negative or AB negative, which is really, really rare, or O neg, right? That's your blood type. And you were to marry a man who was RH positive, okay, and y'all had a child, there's a chance that your baby could be RH positive, which would mean that on your first pregnancy, there really wouldn't be an issue, and we're going to get into why, okay? So the RH comes from rhesus monkeys, because that's when they first discovered this particular blood grouping system, okay? So now going on talking about RH, hemolytic disease of the newborn. That's what we're talking about, right? Because, okay, so the mother's RH negative, the father's RH positive, the mother gets pregnant, the baby's RH positive, okay? So now, it doesn't typically cause any kind of issues in the first pregnancy. Why? Because there's not an exchange of that blood from the mother to the baby until delivery, right? that placenta separates and some bleeding occurs, the mother can be exposed to the Rh positive blood of the baby, okay? And then she can develop antibodies, right? Now, that's not an issue right then, right? But now she has that antibody to the Rh, right? So now she gets pregnant with the second child. It's a possibility that child could be Rh positive as well. So now she has an antibody against it. She attacks the cells of the baby, okay, of the fetus. And then there's a reaction, and there's a condition that results from that, and it's called hemolytic disease of the newborn, HDN. But it's preventable, right? And I tell people all the time, when you decide you want to have children, if you can do it like this, and I realize that's a perfect world and it doesn't already happen like that, because you can prepare your body for pregnancy by making sure you're eating healthy, you're getting exercise, you're ready, right? You want to be as healthy as you can when you decide you want to conceive a child. And I know that's a perfect world, right? But it would be nice if we could do that, right? But the second thing that we can do is the minute we find out we're pregnant, we want to do our prenatal care, right? We want to get with a physician as quickly as we can, so that not just taking prenatal vitamins and checkups, but making sure like things like this are not occurring, or that we can take care of them as quickly as possible, because it's very preventable. You can administer the RhoGAM shot to the mothers, and it's the Rh antibodies during and after the first pregnancy to prevent an issue in your second pregnancy, okay? Okay, so just remember, if the mother's positive and the baby's positive, they're both the same, so nothing's going to happen, right? Again, same thing. If the mother's negative and the baby's negative, right? Nothing's going to happen because they're both the same, right? Okay, so would a hemolytic disease of the newborn affect the first child of a mother who is negative and a baby who is positive? No, right? We talked about that. The mother would not produce any Rh antibodies until after she gave birth to that baby, right? Because she's not going to be exposed to those cells until right near the end, and then it's going to take her some time to build up those antibodies to it. So consequently, it usually does not affect that first birth, right? And then, true or false, when a negative mother receives a shot of RhoGAM, the physician is actually giving her Rh antibodies, and that is true. So use this slide to see if you are totally understanding this and can apply it, okay? So leukocytes. Now we talked about RBCs, red cells, erythrocytes. Now we're going to talk about leukocytes, or white cells, right? So there's two major groups of leukocytes. You have your granulocytes and your agranulocytes. Again, guys, this is where that medical terminology comes in handy, okay? Agranulocytes means without granules in the cytoplasm, and granulocytes, of course, means with granules in the cytoplasm, okay? So let's go through some of these so we can identify them when we see them, right? Okay, so neutrophils make up the biggest percentage of white cells in your bloodstream. It's about 50 to 70% of the cells in your bloodstream are neutrophils. Okay, so when we stain them, because we're always going to stain these slides before we look at them to identify our white cells, right? Because that's how we identify them, about the way they take on stain, right? So the granules in the neutrophils are much finer stain. They stain a bluish color. You're going to see that nucleus in the center there. You see that top picture. You see all the red cells around it. And you see one little platelet there, and then you see your white cell, right? That's a neutrophil up there at the top. Because you can see some fine granules in that cytoplasm, and you see the nucleus, and it has little lobes to it, right? They have at least 2 lobes, sometimes more, and we call them segmented neutrophils, okay? Because you see the little segments or lobes, right? You usually see an increase of neutrophils if these shoot up on a differential. Then that usually indicates the possibility of a bacterial infection. The next we're going to look at is that middle slide. These are eosinophils. They make up about 1 to 4% of your total white cells. Their granules stain. They're bigger, for one thing. You see them more prominently. And they stain an orangish red color with acidic dyes. They have a nuclei, and they're usually 2 large lobes. Sometimes it's hard to make them out What you really shows you it's eosinophil is those red-orange granules that are kind of real pronounced. They're not as fine and kind of backgroundish in the neutrophils. They really show out in those eosinophils. You usually see an increase in eosinophils in allergic reactions. You have hay fevers, allergy season, or in certain types of parasitic infections. Okay, and then last down there at the bottom is basophils. And the common thing between eosinophils and basophils is those granules in the cytoplasm really show out. The eosinophils there with that really orangish red, and the basophils with that really dark purple, almost black looking. Okay, and then a lot of times, if you can make it out on top under the granules, it has an S-shaped nuclei. And it makes up about 0 to 1% of the leukocytes in your system. Okay. So now we talked about granulocytes, the ones with granules, right? Now we're going to talk about agranulocytes, the ones without. And we got two pictures here. The one at the top is your lymphocytes. They make up the biggest majority of those, right? 20 to 35%. It's also the smallest WBC. You'll notice, and you can from this picture, and even more if you were looking at it under the scope, it is much, much smaller than that monocyte. So, and even though there's more of them, they're smaller. Also, it's very little cytoplasm. It's a real big round nucleus and a very small amount of cytoplasm, okay, with these lymphocytes, and that helps you identify. These are seen, you see increase of lymphocytes with viral infections, okay, and with mononucleosis, okay. They produce antibodies. Lymphocytes produce antibodies, okay. So, and then at the bottom picture, we have the monocyte. It makes about 3 to 8%. They're the largest of the WBCs, and they have that one single kidney bean-shaped nucleus, and you'll be able to see it. Looks just like a little kidney bean in that nucleus, and it's much larger, has a little more cytoplasm than that lymphocyte. But you guys are going to have to be able to identify these when you see them, okay? So, still talking about leukocytes, right? WBCs are formed in the bone marrow for the most part, sometimes in lymphatic tissue. Okay, your normal range of to make sure that you fall into a normal range for your WBC is 5 to 10,000, right? A low WBC usually indicates a viral infection, and a high WBC usually indicates a bacterial infection, and a very, very high WBC can indicate blood cancer or leukemia, right? Okay, so that's why I really think a CBC is an extremely useful tool for physicians, because we want to diagnose this. Because a lot of times these viral infections and bacterial infections, as far as their signs and symptoms, they mimic one another, but we don't want to overuse our antibiotics by giving it to people who have viral infections. And one way for us to help distinguish between viral and bacterial infections is to run a CBC and to do a differential. So what is a differential? You'll see CBC with a diff ordered quite a bit. We draw it in that purple top or lavender top tube that has an additive, which is EDTA, which is an anticoagulant, because we don't want that blood to clot, right? Because we're counting the cells, and how would we be able to count them if they clotted, right? So differentials can either be done through hematological instruments, these special instruments that can do a differential, are printed out along with your white cell count, your red cell count, your platelet count. There's some other indices that are on there. Your mean corpuscular hemoglobin volume, things like that are all listed out in this printout from your hematology instrument. But a lot of times a doctor will also order a manual differential, okay? So you'll get the printout from the CBC, from the instrument, you make your slides, you stain them, you put them under your microscope and you look at them, and then you'll do a count of your white cells. You'll make an estimate. Sometimes your platelets, you'll look for any oddities in your red cells and things of that nature, okay? So WBCs are also specific cell surface antigens that are expressed at certain times during development, right? The antigens can be identified by the laboratory method. So they have these cell surface antigens that are on these cells, and we can use flow cytometry or monoclonal antibodies to be able to tell how immature these WBCs are. Is it a ruboblast? what stage is it at, and it helps in cases of leukemia, because you got to realize that when you're throwing out large sums of white cells out into your bloodstream, whether it's a bad bacterial infection, maybe you become septic, and your bone marrow is working overtime to get out as many white cells as humanly possible, sometimes it's going to start throwing them out when they're not all the way mature, right? And so in cases of leukemia where your white count is extremely high, you really will see a lot of these immature cells out there in the bloodstream that shouldn't be out there. And the more immature they are, the more we can identify this as a leukemia. So there are all those tests that can identify from those cell surface antigens which stage that white cell is at. So we're not going to do in person right now mock differential count, right? We will do this in lab though. But just to go through this so you will understand how it works, you could do it on a sheet of paper, or down here in the bottom right hand, that's a cell counter, right? Across the top, you can't see that. There's going to be a picture of each different cell that you will see. And then each time you see that in your field, you will click off each one, and at the end, you're going to get a percentage, right? So you're looking for lymphocytes, monocytes, neutrophils, basophils, and eosinophils, okay? So I'm going to show you these first few slides. I'm going to tell you what they are, okay? So what we're seeing right here is what, okay? We see this segmented, multi-lobed nucleus with little granules in there that stain kind of a blue color, right? That is what? A neutrophil, right? And right above it, that little small little speck of blue right there above it, that's a platelet. And then, of course, we see these red cells with this kind of white center, right? That's why I call them donut, because it's that biconcave, and that's how it appears as that light is shining through it, because that center part is just much thinner, so the light comes through it better. So those are your red cells, okay? I'm going to go through a few of these, and then I'm going to click through them pretty fast after that. And I want you to be able to pause them, take a sheet of paper out, and see if you're starting to get the hang of being able to identify them by sight. Because again, this will be on a test. So again, here we go, right? Multi-lobed. We got 1, 2, 3, 4 segmented lobe. We see little granules in the cytoplasm in the back. It's a bluish color. What is it? It's a neutrophil, right? And of course we would see 2 neutrophils back to back, because what are the majority of the cells in there? Neutrophils, right? What about this one? Okay, we got small cell, big nucleus, just one big round nucleus with almost no cytoplasm. Do you remember what this one is? A lymphocyte, right? We see 2 platelets over there. There's our red cells, right? This is a lymphocyte, a small cell, really big nucleus with almost no cytoplasm, lymphocyte. Okay, what about this one, right? We don't really see any granules. See how smooth that cytoplasm is? There's no granules in there, right? We got a really big cell, right, with a kidney bean-shaped nucleus. What is it? We talked about this one, right? The largest WBC, monocyte, right? There's our monocyte. So again, see those granules in that cytoplasm? Segmented lobe, multi-lobes. What do we got? Neutrophil, right? Big nucleus, really small cell, hardly any cytoplasm, lymphocyte, right? And of course we see more of these, right? Because there's a lot of them in here. We see that granules in our cytoplasm. Segmented neutrophil, right? This is a neutrophil. 3 more neutrophils, right? We see those granules in the cytoplasm. Note, see how smooth that cytoplasm is? You don't see those granules in there, but do you see how there's hardly any cytoplasm at all? But there's a really big nucleus right there in the center, right? What is this? It's our lymphocyte, right? Again, really big cell. See how much bigger this one is than our previous cell? Because we know that lymphocyte is the smallest one, right? The smallest one with almost all nucleus, hardly any cytoplasm. And here we go to this one. It's obviously much bigger. It's got more cytoplasm. Of course, we don't see any granules in that cytoplasm. Kidney bean-shaped nucleus. This is what? It's our biggest cell, right? It's our monocyte. So now I'm going to click through these okay? You can pause it, but I'm going to click through them quickly. You should get out a sheet of paper and you should test yourself to see if you're able to identify these, because you will have to do it on a test. Okay, now this one we haven't seen yet, right? But we see those granules in that cytoplasma, right? We see them, and they're reddish orange color. Which one did we talk about that was a granulocyte, that the granulars were much more pronounced, we could see them a little better, and they were orange and reddish? It was our eosinophil, right? This would be our eosinophil. You can name this one. You've seen it many times before. You've seen these many times before. Okay, here's one we haven't seen just yet. We went over it though, right? We see those granules in there, and they jump at us at that dark, dark purple and blue. What's this one? The basophil, right? That reddish orange that we saw before, that's our eosinophil. This dark blue, big black almost granules in there, this is our basophil. Orangish and red, right? What is it? Okay, so these were only 25 cells, okay? But in a real differential, you would count 100 cells, okay? And it's reported as the percentage of each type of white cell. That's why you had that percentage, right? You fall into that normal percentage. Okay, so remember, I'm going to give you pictures of each one of these WBCs, and you will have to identify them, okay? So make sure you use these PowerPoint slides as ways to test yourself and make sure you're able to spot them when you see them. And remember, guys, you know, phlebotomy is a great job. It's a key, intricate part of the healthcare team, but I always love to see people continue their education, to go on and get a 4-year degree. You can become a clinical laboratory scientist. You can go on to become an LPN, a respiratory therapist, a nurse, a medical assistant. You never stop learning and growing. And if you have any inclination that you want to go further and become a clinical laboratory scientist, then you will definitely need to start learning these cells now, not just for this class, but for your future. Remember, these are all the ways that we'll know. It's a telltale sign to help the doctor with his diagnosis, his treatment, right? Whether you're bacterial infection or viral infection, or you could have allergies or a parasitic infection, right? Okay, so here's some ways for you to be able to look at the information you're obtaining in each one of these boxes and test your knowledge to see if this stuff is clicking with you guys. I can't express to you enough how important using these as a study tool is for you. Okay, so on this one it talks about box B. Is it a normal differential? Which of the WBCs in box B are abnormally high, and what condition does it indicate? So look at that box, find the one that is abnormal, and then tell me what it could indicate in our patient. Okay, so we talked about erythrocytes, our red blood cells. We talked about leukocytes, our WBCs, and now we're going to move on to thrombocytes, right? Thrombocytes are our platelets, and they're fragments of a much bigger cell called a megakaryocyte, okay? The normal range for platelets is 250 to 500, okay? And platelets help in the clotting process, right? They transport chemicals that are needed for clotting, and they form that platelet plug, right? That slows blood loss. Abnormalities in either the quantity or the quality of your platelets can result in bleeding disorders, which are detected by bleeding time test and the platelet count, okay? Thrombocytopenia, and this is why, sorry. Okay, again, sorry guys. So thrombocytopenia, this is why medical terminology is so important, is a low platelet count, right? Okay, and it's treated, one of the treatments for it is by platelet transfusion, okay? And then thrombocytosis is too many platelets. That's like that trying to keep hemostasis, right? You want to keep just the right amount, normal range of platelets. If they get too low, then we might not be able to clot when need to. And if you have too many of them, it could cause blood clots to form, right? You could clog up blood vessels and then cause death in your brain tissue, in your heart tissue, or wherever, right? Another reason that I always tell y'all that the proper IDing of your patient is vitally important. You must ID your patient. Then you must identify yourself, where you're from, what you're there for, right? We have good customer service. Okay, but you also want to do a medical interview, and part of that medical interview is asking patients if they're on any medication, because if they're on any kind of anticoagulation therapy, and we talked about what anticoagulation means, right? It stops coagulation, right? Well, that's important when you're drawing blood because you're sticking a needle in their arm, right? And if they're on some aspirin or heparin or Coumadin or any other anticoagulation therapy, then it may take them longer to stop bleeding. You will need to know that so that after you draw their blood, you hold pressure for longer. That's one of the reasons that medical interview is so important. Okay, now on to the plasma. This is the liquid portion of our blood, right? We talked about some of the stuff that's in there, the solutes. And there's an error on this slide. Y'all make sure you notice this. Plasma is composed of 90% water and 10% solutes, not the reverse, okay? 90% water, 10% solutes. Sorry, I'm gonna fix that today. Okay, so the plasma portion of your blood is your liquid portion, right? So if we drew it in this purple top tube we see here, and a purple top tube is mainly used for hematology studies like CBCs and hemoglobin to hematocrit. It has an anticoagulant in it, right? Because we don't want those cells to clot up because we're looking at those cells, right? And it's delivered to the hematology department. It has that EDTA in it, which is an additive. Anything put in a tube is an additive, and it's an anticoagulant, right? And if we took that tube and we spun it down, the stuff that would be at the top would be plasma, right? So in that plasma, it's 90% water, and then it's 10% solutes. And those solutes are things like glucose, fats, hormones, clotting factors, and it's all those things are dissolved in that water, okay? The WBCs and platelets, they form this really thin white layer right above the RBCs when it's spun down, and we call that the buffy coat. So you got the plasma, then you got that little thin white layer, which is your WBCs and your platelets. It's called the buffy coat. Then right under that, it would be our cells that are packed down, right? Okay, the pH of your blood describes the degree of acidity or alkalinity in your blood. And if the normal blood pH is 7.35 to 7.45, and as you can see, that's not a big window there, okay? We really need to keep this pH in that normal range, okay? So if a blood specimen that does not contain an anticoagulant, and if you've watched the order of draw video, which I hope you have, it's vitally important to not only know the order of draw, which takes going over it repetition to have that rogue memory to memorize the order of draw, but it's important to know why. Okay, so here we have a picture of a red top tube, and in our order of draw video, you learned, and you will learn that in lab as well, and we'll go through it in here in lecture as well. A red glass red top tube has no additives, okay? So there's nothing in there to prevent it from clotting, so it will clot. And depending on the type of tube and the additive in that tube, and this plain glass red top tube is going to take the longest to clot. It could take up to an hour for this blood to clot. That's why we don't really use it a lot, because it interrupts turnaround time. Because you have to wait an entire, sometimes almost an hour for it to clot, and you cannot spin it down. You can't put it in the centrifuge and spin it down until it is fully clotted. Okay, and we'll get into that later as to why. Okay, so you drew it in a plain red top tube, it clotted, you spun it down. What's at the top? Okay, it's not plasma, now it's serum. Okay, and you don't have that white buffy coat any longer. Okay, and I'm going to talk about why. And now you have this clot down at the bottom. Well, the reason you don't have that white buffy coat is because we allowed it to clot. Platelets are involved in the clotting process. Fibrinogen, right? Other things. And they all get into this nasty clot down at the bottom with red cells, white cells, platelets, fibrinogen, and everything is down there in the bottom. Okay? So you don't have the buffy coat because all those things got used up in the clot, right? So the difference between serum, which we're looking at right now, and plasma, they're essentially the same except for the clotting factors, okay? The alga protein fibrinogen, the platelets, and all the cells are all now in this massive clot down at the bottom. So you couldn't use this for cell morphology because the cells can't run through any machine. They're not liquid now. They're just this big, massive clot down there. They clog the machine up, right? And if you're looking to test for clotting factors, you can't do that either because they're used up, right? All the cells are used up and all the clotting factors are used up. So the difference between serum and plasma is plasma still has the white cells intact, still has the red cells intact, still has the platelets, all the clotting factors, fibrinogen, all that is still present in a tube that has plasma in it. They do not in a tube that has serum. And y'all are going to be tested on this. What's the serum? What's the plasma? What tubes are they in, right? If you have a tube like a green top tube or a purple top tube and you spin them down, that's going to be what? Plasma, right at the top, because it has an anticoagulant in those tubes. But a red top, an SST tube, right? Those, plastic red, glass red, those have no anticoagulant in them. So once they clot and you spin them down, that's going to be serum at the top, okay? Okay, we talked about homeostasis before, the steady state of your body, right? Hemostasis, right? Blood, hemostasis. This is a very complex series of processes in which platelets, plasma, and coagulation factors all work together and interact to control bleeding, while at the same time maintaining circulating blood in a liquid state. This gets deep, guys. It's very complex, and you're going to have a specific study sheet to help you be able to see the cascade of it all and how it works, okay? So you want to think about what is the difference between homeostasis and hemostasis, right? Again, another reason why medical terminology is so important, right? Homeostasis is a steady state condition of your body as a whole. Hemostasis is a steady state condition of the blood, right? Specifically, the ability of blood to keep the blood in that liquid state while controlling excessive bleeding at the same time. So let's go through the phases, okay? Because we have about 5 phases of hemostasis and coagulation. Okay, so in that first phase, we talk about vasoconstriction phase, right? Because when you cut yourself and you're starting to bleed, the first phase of this whole thing is your vessel walls decrease your blood flow, right? I mean, think about it. We're bleeding. Oh my God, let's constrict it down to preserve that blood so not as much of it flows out, right? So the second phase is that platelet phase, okay, where the platelets clump together to form that platelet plug. And we also know they release some chemicals and things of that nature as well, right? And then that third phase is the coagulation phase. Okay, y'all, there's a lot involved in this, okay? And we will go over this with another handout about the cascade of it all, okay? So the coagulation phase is where calcium and the clotting factors cascade to form a fibrin clot, right? The coagulation phase is divided into two systems, intrinsic and extrinsic, right? Something internal or something external, right? Because when you think about the extrinsic system, this is when coagulation factors and calcium are released from damaged tissue, right? And it lists out the factors that are involved. And the coagulation test for this system is the PT, the prothrombin time. So extrinsic, think about tissue damage, okay? Something happens to you, you have some type of tissue damage, it's involving these factors, okay? And that's what the extrinsic tissue system is about. But it still involves the coagulation factors and calcium, okay? But if we're looking for this specific set of circumstances, we're going to order the PT, okay? The intrinsic system is, again, we're looking at coagulation factors and calcium are used from the bloodstream to form the clot, right? And it involves a multitude of, I'm not going to sit here and read every one of these factors out to you guys, but it involves all these. And the coagulation test for this system is the partial prothrombin time, the PTT. Now, if something's going on and the doctor has no idea what's going on, right? Or you got multiple things going internally and externally, then he may order a PT and a PTT to try to narrow down what could be causing the issue, okay? So the fourth phase is the clot retraction. That's the phase where the clot retracts or gets smaller and pulls the sides of the blood vessels together, right? Because we want them to pull together and close up and heal, right? And the fifth phase is the clot destruction phase, where the fibrolysis, which is enzymatic destruction, occurs, okay? Okay, it destroys in an enzymatic way, it destroys that clot, okay? And now you'll notice, as like if you have a scab there, right? If you pick at it too early, it'll just start bleeding again. Then it'll form that platelet plug again, and then it retracts in and retracts in and retracts in until eventually it's pulled all that that vessel walls together. It's healed underneath there, right? And so that can just fall off of there. That fifth phase has occurred. It has destroyed that clot, and then, you know, that you move on through those phases, right? So the thing we need to take away from here is this is a very complex process, right? And it has 5 phases: vasoconstriction, platelet, coagulation, then you have intrinsic inside the body, right? Extrinsic outside the body, then you have clot retraction, and then clot destruction. So now we're going to talk about disorders of the cardiovascular system in the hemostatic process, right? Because, okay, guys, this is what it's all about, okay? You are going out there as a phlebotomist to draw blood, to run laboratory tests, to try to diagnose, monitor, and treat patients. So you need to understand the disorders of the cardiovascular system and the hemostatic process, so you will know and be able to speak intelligently and understand why you're drawing blood, why you're drawing it in certain tubes. All this stuff goes together so that you will totally not just be able to do a job, but understand why you're doing this job, okay? So we're going to start with disorders in the process of hemostasis, right? Okay, hemophilia. I'm sure y'all have heard this before. It's an inherited condition where excessive bleeding may cause death. You see, they talk about, you know, they don't want to fall down or hurt themselves, and they'll get, they had an issue early on in the AIDS epidemic. A lot of hemophiliacs were getting AIDS because they got a lot of factors, you know, that they took regularly to help control their hemophilia, and that was a type of blood product. So early on, we had a lot of cases of hemophiliacs getting AIDS due to their blood products they were receiving. So DIC, right? DIC is a condition where small blood clots are widely distributed in the body caused by a hyperactive clotting process, using clotting factors up and causing excessive bleeding. Okay, This is I've seen it in the hospital a couple of times. It is not pretty. And even though people can survive DIC, I've personally, in all the cases I've ever seen of DIC, I've personally never seen anyone survive it. Okay, it's very serious. Okay, very serious. Okay, so moving on to the lymphatic system now. Okay, what is the lymphatic system? Okay, it maintains a fluid balance. It provides immunity. Okay, it distributes nutrients and hormones. It removes waste and absorbs fats from the digestive tract. Okay, so you see people have lymphatic problems, and they may have really a lot of swelling in their ankles and their feet and their lower legs because that lymphatic system is not able to do its job. So it's not able to remove that waste and that fluid, so that fluid pulls up down there. But it's also vital in your immune system, okay? So what are some common blood tests that we do, laboratory tests? So tests such as a CBC or an RBC count, hemoglobin, hematocrit, okay, differentials, platelet counts. We got PT, PTT, fibrinogen, okay? All these tests are performed in the hematology or coagulation part of the laboratory. And 99.9% of the time in most laboratories, coag is going to be housed in hematology. So if you go out and draw a CBC and a PT and a PTT, you're going to have a purple top and a blue top, and you're going to take those tubes to the hematology department, okay? Blood groups and type, so group and type, cross-match are done in the immunohematology or blood bank portion of the laboratory. Then you have serum or plasma constituents such as calcium, glucose, potassium, all our panels like thyroid and chemistry panels. All of these are done in the chemistry part of the laboratory. So if you drew a green top tube to do an electrolyte panel, or if you drew an SST tube to do a thyroid panel, you would take those tubes to the chemistry department of your laboratory. So what are the components of a CBC? Guys, you're going to need to know this. This is going to be on a test, okay? What are the components of a complete blood count, a CBC? You're going to have a WBC count. You're going to have an RBC count. You're going to have a platelet count. That should be easy, right, guys? What are your three cells? White cells, red cells, and platelets, right? So a complete blood count, you know right off the bat, it's going to do those three, right? Then we talked about that differential, right? The instrument can print you out a differential. You can do a manual differential. A differential, whether it's from the instrument or manually done, is always included in that complete blood count. We want to see if you have elevated neutrophils. Do I have a bacterial infection? Do I have elevated lymphocytes? Do I have a viral infection? Do I have elevated eosinophils? Could I have a parasitic infection or an allergic reaction to something? Okay, so three cells, right? White cells, red cells, platelets, and our differential, okay? And then we always get the hemoglobin and hematocrit. Do we having some kind of anemia going on due to blood loss or some other issue, right? And then your RBC indices, okay? These are the things that shows the mean corpuscular hemoglobin concentration inside those cells, how much hemoglobin's in there, and others, okay? So if I ask you to list out on a test, and I will, what are the components of a CBC? The first thing you should think of is complete blood count, okay? My white cells, my red cells, and my platelets, right off the bat, right? Then I know I'm going to always get a differential, at least off that instrument, right? Then I need my hemoglobin and hematocrit. Do I have any kind of anemias or bleeding going on? And then last but not least, your RBC indices. So make sure you know this and go over it, okay? So what type of specimen is used for hematology tests? Whole blood, right? How can we count cells? How can we count how many white cells are there? How many platelets are there if it's not whole blood, okay? And so hematology tests are usually done in a lavender or purple top tube that has an additive in it, which would be an anticoagulant to prevent that blood from clotting, so you could run that test on whole blood. They never spin it down. It's always kept whole. And if you've ever walked into a hematology department, you might see a little rack over there that's going back and forth very slowly, inverting that tube. And there'll be a bunch of purple tops laying on there because it's keeping that blood mixed together until you get ready to run it through your instrument, because you want to make sure it's inverted and you don't shake it. You just want to invert it gently to mix that blood, okay? So, of course, we're not going to be able to do this in person. We will do this in lab, but just to go through it, because I think it's good for you to understand it and see it so you can start visualizing. So when you sit down to do it, it is better. But you can do this at home with your children or your significant other or someone, your mother, father, somebody. Go find somebody and see if you can find their pulse, right? You can even try it on yourself, okay? So finding an artery, the radial and ulnar are the most commonly used, and they're in your wrist. So you would turn your hand over, take your finger. You never want to use your thumb because there can be a pulse in that thumb. Take your finger, lay it over that area that we talked about. It's right there, okay? And feel for a pulse. And then you want to watch your watch, and you want to count each beat in an entire minute, and then that would be your pulse. So here again is one of our clinical thinking slides that you should use as a way to see if this stuff is clicking and as a study tool for you to study for your test. You can pause it here and work through these. So, we're at the end of chapter 7, okay? So again, you should have already watched the PowerPoint presentations for chapter 1, chapter 6, and now you will have watched chapter 7. You should have read chapter 1, 6, and 7 in your textbook, phlebotomy handbook. You should have filled out your chapter reviews that are in your black binder for chapters 1, 6, and 7. If you have done that and then take your chapter reviews and study them, you will make an A on your first test, I can assure you, if you're willing to put in the time that you need to be able to get this done. And as I told you in the beginning, once you finish this chapter, you should be able to trace that blood flow through the heart. Remember, the inferior and superior vena cava into the atrium, right? The right atrium. You should be able to locate and name the veins and arteries most commonly used for phlebotomy. And you should be able to define and explain hemostasis versus homeostasis, right?